Hai, Assalamualaikum. Sekejap ya. Mana tadi? Sorry. This one. So previously I have recorded the meiosis one. Oh, sekejap, I nak silent TV. Okay, so but the meiosis one video was not complete. Why it was not complete? So today I'm going to continue once again with this topic, meiosis. Okay, so I will review fast forward about the meiosis one, and then we'll continue straight to the meiosis 2. Alright, so previously I tak tahu how to use this app, so now I dah tahu dah cara nak guna apps ni sambil record. Okay, sambil I explain to you this PowerPoint. Okay. So cycle kita dah tengok. Okay, division. So directly goes to our lecture previously, the meiosis kan. Kat sini kita stop dari tu. So a little bit revision of meiosis. Okay, so meiosis reduces the number of chromosome sets from diploid to haploid. So this one that you must already understand the concept of meiosis. Okay, we need to reduce the number of gametes, number of gametes lah. Okay, sepatutnya gametes memang haploid, dia tak boleh diploid. And then once again, dia nak reducekan daripada mana ke mana? From 46 menjadi 23. Okay. So meiosis kena reduce numbers of chromosome sets from 46 to 23. So meiosis have two steps. We have meiosis 2 and meiosis 1 and meiosis 2, and both are preceded by the replication of chromosome. But interface cuma berlaku waktu meiosis 1, sebelum meiosis 1 sahaja. Right. So after completed meiosis, we will have 4 daughter cells. Okay, and different with the mitosis, we only have 2 daughter cells. But meiosis we have 4 daughter cells. And the setiap sel tu dia adalah N. Okay, dia tak ada 2 N, dia cuma N saja. Each final daughter cell has only half as many chromosome as the parents. Okay, sebab ini semua berlaku, meiosis only occur at gametes, which are the sperms and eggs. Okay, or eggs. Okay, kita nak move. Okay, sini ni pun I dah terang hari tu. Stages in meiosis, we have two stages. Meiosis 1 is the reductional division, which during this meiosis 1, homologous pair will be separated. But in meiosis 2, we call that as a equational division, which is we want sister chromatid to be separate. Okay, so you can ingat waktu first meiosis 1, kita nak separatekan apa? Homologous chromosome. Okay, we want to separate this homologous chromosome during meiosis 1, but in meiosis 2, we are separating the sister chromatids. So after meiosis 1, daripada satu sel dia menjadi 2 sel. So satu sel, okay, and then boleh jadi 2 sel. Sorry lah, saya punya lukisan ni pakai tracking pad ni jadi macam ni. Okay, but after meiosis 2, we can produce 4 daughter cell. Okay, so at the end kita ada 4 lah sebenarnya lepas habis meiosis 2. Okay, alright, so these are the drawing that I draw you earlier. Okay, so basically this is after meiosis 1. So after meiosis 1, so kat atas ni, okay, lukis lagi sekali. Yeah, so sini dia masih lagi 2N right, so this is still 2N. Complete meiosis 1, dia dah jadi N dah. Walaupun di dalam bentuk homologous chromosome, it already N, have been reduced the N number, and further reduced for meiosis 2, but the cell number is still N. Cumanya, sini ada 2 sel, in this one we have 4 cell. Okay, so first division, meiosis 1 separates homologous chromosome. Kena ingat, you need to remember this. Second meiosis 2, it separates the sister chromatids. Okay. So division of meiosis 1 is the separation of homologous chromosome. We have these phases. Division of meiosis 2 is the separation of sister chromatid. I have repeat this a million times. Meiosis 1 kita cuma nak separate homologous chromosome. Apa itu homologous chromosome? I need to recall back what I told you earlier. And then in meiosis 2 we need to separate the sister chromatid. Okay? So this is an overview of meiosis. Okay. Okay, as you see here, okay, meiosis 1 separate homologous chromosome. These are original cell, divide into two cells with homologous chromosome in each cells. And then kalau you tengok ada what different colors of chromosome. Ada the blue sets and the red set. Yang warna merah dengan warna biru kan. So these different colors means that it came from parents. Satu daripada mak, one from the mother, one from the father. Ataupun kita panggil the paternal or maternal set. Okay, maternal from mother, paternal from father. Okay, so dalam gametes memang macam ni. Okay. And then meiosis 2 kita nak separate sister chromatids. Okay. At the end we will have 4 daughter cell and each of the daughter cell have haploid number and different genetic material from the parent cell. This is the parent cell. So bila dia melalui meiosis, dia dah ada combination of genetic material from parents. Okay, these are against the first meiotic division, second meiotic division. Okay, so interphase is the resting stage for before meiosis 1 start. Okay, so antara meiosis 1 ke meiosis 2 kita tak ada interphase dah, kita cuma ada interkinesis. Dia dah rest seketika tapi no DNA duplication, dah tak ada duplication dah waktu tu. Alright, so I just go fast forward this one because I have already mentioned it. I will continue with meiosis 2 directly. Alright, so kita dalam meiosis 1 we have certain events that important. For example, in prophase 1 we have the what's that called? Synapsis kan? Synapsis and crossing over waktu prophase 1. And then waktu metaphase 1 we have the independent assortment. Okay, but for prophase 2, okay, kita dah masuk meiosis 2 sekarang ni. When we go into meiosis 2, the step is similar like mitosis. It's just that we call them as a prophase, as the meiosis 2. Okay, so that's why it said here meiosis 2 is very similar to mitosis. Okay, prophase, for example here prophase 2, chromosome will start to condense again. Okay, each homologous chromosome consists of two chromatids and attached at a centromere. And we do not have synapses here. No synapses occur. Synapses do not occur and the nuclear membrane and nucleolus disappear and the spindle fiber will form. The step is similar like the mitosis. It's just that this time we want to separate the sister chromatid lah. Okay. Each metaphase 2, each of the chromatid line up in the equatorial or metaphase plate of the cell. So semua beratu kat sini. Okay. So kalau you perasan dah ada different genetic material kat sini. Dia dah mengalami crossing over lah, exchange of the genetic material. Okay, and the two sister chromatid which is we dah bagikan kat tengah macam ni. Two sister chromatid of each chromosome are not genetically identical. Then the kinetochores of sister chromatid attached to microtubule extending from opposite poles. Okay, so kinetochores will attach to the microtubules and microtubule will pull apart this sister chromatid to the different opposite poles. Okay. And next, after metaphase, so kata kunci untuk metaphase ni adalah dia line up dekat metaphase plate. Sama ada either meiosis 1 ke mitosis ke meiosis 2 ke, semua waktu metaphase dia akan line up dekat metaphase plate. Alright, next we move forward to the anaphase 2. So in anaphase 2, the sister chromatid will separate. Waktu ni lah sister chromatid akan separate and the sister chromatid of each chromosome now move as two newly individual chromosome towards opposite poles. So dia dah bergerak ke arah pole yang berlainan. Okay, satu kutub utara, satu kutub selatan. And then proceed with telophase 2 and cytokinesis. Chromatids reach the opposite poles and the spindle will disappear. So bila dah sampai dekat opposite poles, spindle fiber pun akan hilang balik lah sebab fungsi dia dah selesai. And then the nuclear envelope and nucleolus will start to reform in each daughter cells. After telophase 2, it follows by cytokinesis. Thus, meiotic division will produce 4 daughter cells that have the variation in genetic material. So sekarang ni fungsi meiosis tu kita nak pelbagaikan genetic material inside the gametes. Okay, so kita tak naklah adik-beradik kita sama macam, sebab tu that's why our siblings do not have exact copy like us. This is because of meiosis actually. Okay, alright. So origin of genetic variation amongst offspring. This is what I mentioned earlier. Amongst your sibling itself, we have this genetic variation, betul lah? Okay. The behavior of chromosome during meiosis and fertilization is responsible for most of the variation that arise in each generation, and three mechanisms contribute to genetic variation. So we have a mechanism in order to maintain this variation in our genetic material. First is independent assortment of chromosome during the metaphase 1. Okay, independent assortment. Second mechanism is the crossing over bila prophase 1. So I lukiskan lagi kat sini. Sorry, this one lah. Okay, this one occur independent assortment waktu metaphase metaphase. Tapi I tulis metaphase I satu je lah. Okay, crossing over waktu prophase 1. Random fertilization, I think it occur during telophase 1 kot. Nanti kita tengok Okay, sorry I tak ingat yang tu. Alright, so first we look at this independent assortment. Okay, during independent assortment, saya kecilkan sikit. Sekejap I remove. Okay. Independent assortment of chromosome. Alright, supaya tak tindih dengan muka. Alright, homologous pairs of chromosome orient randomly at metaphase 1. Okay. So kita tahu independent assortment berlaku waktu metaphase 1. Okay. In independent assortment, each pair of chromosome sorts maternal and paternal homologous into daughter cell independently of the other pair. So what does it mean here? Apa yang dimasukkan kat sini adalah waktu independent assortment, okay, waktu metaphase 1, homologous chromosome tadi akan beratur dekat metaphase plate tu secara random. Kita tak boleh tentukan paternal set belah atas semua, maternal set belah bawah, tak ada. Dia beratur secara random lah, secara random. Okay. So each pair of chromosome sorts maternal and paternal homologous into daughter cell independently of the other pairs. So, contohlah kita ada 3 set, 3N dalam satu sel tu. So I lukis sikit kat sini. Okay. For example, we have 3 sets of chromosome. I lukis different color lah. Pink dulu. Okay. So we have, mana dia tulisan? Okay. Lah bukunya. Ada size, different size kan? Ada yang kecil, ada yang besar, ada yang panjang. Remember back that I said earlier. Okay. For example, ni dalam satu sel lah. Okay. Ini contohnya daripada mak. Okay. Now we are looking the paternal set from father. Kita letak sebelah-sebelah. Maksudnya sekarang ni dia dah pair kan, dia punya ikut size dan kepanjangan lah. Okay. Sorry kalau tak sama besar. I'm using this tracking pad. No. Sorry. No lah. Okay. Alah. Okay. So, kalau macam ni, dia cakap 3N sama dengan 6 lah. Okay, 1 set, 2 set, 3 set. Okay. So, nak beratur dekat tengah-tengah sel tu, kita lukis pula sel. Maafkan saya. So, if the chromosome nak align at the metaphase plate ni, okay, kita buat imaginary plate kat sini. Okay. So, adakah dia akan beratur semua merah belah atas, semua biru belah bawah? No. Not necessarily. It can happen like that, but not necessarily. That's why I said it is independent assortment. So, it can be bergila-gila lah. Sorry, warna tak sama. Tapi anggaplah ni pink ya. Okay. Maybe belah atas tu pink. Bawah pula pink. Dia kan ada it have dia punya pasangan sendiri kan. So, dia boleh beratur susunan dia tu tak bergantung kepada apa-apa pun. They just sort independently. So ni maybe first arrangement. So lagi satu sel yang lagi, kan kalau dia ada 4 sel, 4 sel lah, 4 daughter cell kan. So maybe first cell it arrange like this. Second cell it arrange another, lagi satu jenis yang lain pula. Sekejap maybe satu lagi kita buat yang baru lah. Ni. Okay. Sorry. Okay. So another cell, kita bayangkan ni sel lain. So mungkin sel lain tu dia arrange atas semua merah, bawah semua biru. It can be like that. So that's why we say that it is independent. Okay. Ataupun it can be like this. 2 merah ke atas, 2 biru kat bawah. Okay, we have kita pakai pattern lah, arrangement. So this is an assortment. So we have different probability. Satu yang macam ni, okay. Another one we can have this kind of ni yang pertama, okay. We can have this first pattern. Ni maybe boleh jadi yang kedua. So kita ada banyak probability lah macam mana dia nak susun themselves at the middle of the metaphase one plate tu. Okay. So you tengok betul-betul dalam notes, dalam manual you. What is independent assortment? Understand carefully. But today I try my best to explain to you. This independent assortment boleh menyebabkan kepelbagaian dalam sel. Okay, during meiosis 1. So we go back to our PowerPoint. Okay. This is what we call as independent assortment. Yang saya cerita tadi tu lah. Okay, cara kromosom tu beratur dekat metaphase plate tu macam mana. Okay. Right. So this is first possibility. Okay. Second possibility. Cuba kita besarkan. Alright. Okay. Awak hang ni. Oh, jam pula. Okay. Possibility 1. Dia beratur macam ni. Comel aja. Biru belah sini, merah belah sini. First, contoh. This is paternal set. The other one is maternal set. Okay. And then, when it's undergo metaphase 2, okay, meiosis 2, it's separate like this. Sama. Sebab dia daripada satu group lah maksudnya. Okay. Allah, jam pula. And then we have this combination all from paternal. This one all from maternal. But we can have another possibility here. So dia beratur tak sama kumpulan. Kalau ni dia sama kan. Ni lain-lain. Dia selang-seli. So we can have this combination. So dalam sel yang daughter cell yang terakhir tu, 4 daughter cell tu, we can have 4, kan kita ada 4 daughter cell kan. So each of the daughter cell may have different combination of the genetic material from father and mother. Okay. Buat ni. Okay. Alah. So, I need to do something. Sorry ya. Sini, tutup dulu lah. Maybe dah banyak sangat buka. Okay. I'm sorry. So, we need to PowerPoint to relaunch. Okay. So, tadi kita sambung dekat. Come on. This one dah. Oh, okay. Okay, sorry. Possibilities, we have different possibilities. So, this is because of the independent assortment. Sebenarnya bukan 2 saja. Ada banyak possibility. Kalau this is 2n sama dengan 4. Kalau kita ada 46 kromosom. 2n equals to 46 kan. So, kita ada banyaklah possibility kita boleh ada kat situ. Macam mana nak kira possibility tu? I think ada dalam manual dia bagi tahu. Possibility during independent assortment. Muka surat 104 Independent assortment refers to the random arrangement. Okay. for a possible arrangement of chromosome during metaphase 1 from an individual that has 6 total chromosome. So tu daripada 6 total chromosome. Kalau kita ada total chromosome kita 2n equal to 46. So possibility nak arrange chromosome dekat metaphase plate tu lagilah pelbagai. Okay. Ni saya ada cara kira je, tapi kita takkan bincangkan kat sini lah supaya you all tak pening. Okay. Second event or second mechanism that contribute to the genetic variability is crossing over. So this crossing over produces recombinant chromosome which combine DNA inherited from each parent. So crossing over begins very early in prophase 1, okay, as homologous chromosome pair up gene by gene, and in crossing over, homologous portion of two non-sister chromatic take trait place. So ingat balik waktu crossing over yang berlaku waktu prophase 1, okay. So waktu prophase 1, dia bertukar genetic material, okay. It change genetic material at synapsis. Oh, besar ni gambar. Sorry. Okay. So it change genetic material during synapsis. So ini adalah kita boleh kata kaedah mekanisme yang kedua, bagaimana sel tu mempelbagaikan genetic material dia. Yang genetic combination yang pertama tadi menggunakan teknik ataupun mekanisme independent assortment waktu metaphase. Now is crossing over yang berlaku waktu prophase 1. Tadi waktu metaphase 1, now is prophase 1. Okay. Oh, no, hilang dah gambar kau. Okay, kita nampak floating. Alright. So this is an example of crossing over during meiosis. Can we enlarge this? Okay, tengok kat sini. So this is pairing of homologous chromosome. Okay, homologous chromosome dah pair dah, and then it change the genetic material at the synapses. Synapses ni dia macam this. Okay, kita boleh kata synapses tu adalah pairing of the homologous chromosome. Sorry. So synapses tu adalah proses nak pairkan homologous chromosome tu tadi. Okay, so bila dia dah pair, so when this homologous chromosome are already paired, it start forming this structure known as the chiasmata. Dia jadi bentuk X macam ni. Okay. So this chiasmata can occur at both side ataupun bahagian bawah saja ataupun bahagian atas saja. Depends. Tak semestinya berlaku dua serentak macam ni. Okay. And then so during prophase dia akan tukar genetic material. So, ambil sikit saja bahagian atas ni. Okay. So kalau you nampak yang biru ambil sikit merah, yang merah ambil sikit biru. Kan? Sikit je dekat sini. Ni tengok ni sel ni. So dia dah bertukar lah ni genetic material, kita dah exchange between the maternal cell and the paternal cell. Okay. So proceed with anaphase 1, anaphase 2, and lastly, daughter cell will have this kind of combination. This is truly from the maternal set. The combination of maternal and paternal, same like this combination but different loci. This one from the pure genetic material from the paternal. For example, we have 4 combination here. Okay. So this is crossing over yang berlaku during meiosis 1. Itu dalam prophase 1 lah. Okay. So kebanyakan yang genetic variation ni disebabkan oleh proses yang berlaku dalam meiosis 1. Prophase 1 dengan metaphase 1. Okay, this one is random fertilization. Alright, random fertilization adds to genetic variation because any sperm can fuse with any ovum. Okay, so sperm ada berapa million kan, berapa juta, banyak. Kita tak tahu which one sperms yang boleh penetrate the ovum. So this is called as random fertilization. Okay, so this random fertilization also add up to the genetic variation selain daripada crossing over dan juga independent assortment. Okay, so ovum, this is unfertilized egg which is haploid egg, n lah, n equal to 23. Sperm also n equals to 23. Tulis sikit. Okay, so this is n equal to 23. So that's why kita bagi di haploid Ovum pun sama. N equals to 23. So when these two gametes, sperm and ovum, fuse together, fertilize together, forming this diploid zygote. So already zygote ni dah jadi 2n equals to 46. Okay. So inside this zygote, we have combination of maternal homologs, which is from this zygote yang warna hijau ni, and paternal homologs from the sperm. Okay. Tapi daripada sperm ayah tu pun tak semua combination of genetic material tu sama. We have different millions of sperms, okay, and we have each month for woman only one yang keluarkan ovum ni lah, okay. Alright. So here we want to differentiate what between mitosis dengan meiosis. Apa beza dia? Okay, you can tengok betul-betul. Waktu mitosis, we have this duplicated chromosome, but this chromosome dia tak ada homologous. Dia tak pair, homologous chromosome tu dia tak pair. It just properly this chromosome line up at the metaphase plate waktu metaphase and then separate normally. Okay, forming 2N. So, kita tengok kat sini. First, yang pertama, yang paling major sekali, okay, we have 2N equal to 6. Di kromosom yang tak duplicate lagi lah, dia bentuk satu macam ni. So, bila dia duplicate, dia akan jadi bentuk butterfly macam ni. Alright, so, metaphase, all the chromosome akan line up dekat metaphase plate. Okay, and then dia akan jadi 2N juga. Sama. Bilangan dia sama sebiji macam parent cell dia. This is a daughter cell of mitosis. Of course, mitosis dia macam we call photocopy, okay, from the original parent cell, photocopy daughter cell, but only have two cell. Cell yang pertama, cell yang kedua, daughter cell. Okay, but different for meiosis, we have homologous chromosome. Sebab kita ada fertilization, fusion of gametes from sperms and from ovum. Okay, daripada gamet jantan dan gamet betina. So, dalam meiosis kita ada homologous chromosome. So, prophase 1, homologous chromosome akan pair up and then exchange genetic material at crossing over. And then waktu metaphase 1 kita akan ada satu lagi event which is the apa nama ni? Independent assortment. So then we proceed with anaphase 1 and telophase 1. Sama juga kita ada sebab waktu ni dia dah jadi n equals to 3. Daripada awal tadi 2n equal to 6. Dia dah divide by half. 6 bahagi 2. 3 lah. Okay. N equal to 3. And lastly, after finish telophase and cytokinesis, kita ada 4 sets of daughter cells, different combination of chromosome. Okay. So, cara disusun kat tengah-tengah ni adalah independent. So, ada 3 event yang menyumbang kepada genetic variation. The first event is crossing over, second event is independent assortment, and the third event is the random fertilization yang daripada sperm tadi. Alright, so important stages in meiosis, we have prophase 1 because we have event of the, event crossing over lah waktu prophase 1. Okay, this is important event you could remember. Metaphase 1, we have chromosome that line up as homologous chromosome. Okay, and anaphase 1, homologous chromosome separate from each other. Sister chromatic remain joined at the centromere. So, bentuk yang macam ni, yang different set daripada satu pair tu, okay, kita panggil dia homologous chromosome. Nama lain dia kita boleh panggil dia sebagai tetrad. Macam ni je dia. Tetrad. Okay. Ataupun bivalent if I'm not mistaken. Okay. So, sebenarnya dalam meiosis, kebanyakan important event tu berlaku dalam meiosis 1 lah. Prophase 1, metaphase 1 and also anaphase 1. So, this question is quite popular. Okay. Selalulah ditanya kenapa, apa beza dia mitosis dengan meiosis ni. So, you boleh bezakan dari segi, first point yang boleh bezakan adalah berlaku dekat mana. Occur in meristematic untuk mitosis and occur in reproductive cell for meiosis. And then second point is the chromosome number. Okay. Sorry. Okay. And then apa yang berlaku dalam prophase? Ada synapsis tak waktu mitosis? Tak ada kan? So basically kita ada berapa point kat sini? 1, 2, 3, 4, 5, 6, 7. 7 point that we can differentiate between mitosis and meiosis. And then daripada 7 ni, at least you remember 5 should be okay. Alright. So this is the end of the meiosis. Okay. So next class nanti I akan masuk this one lah. genetic material, hereditary of our DNA. Okay, so kita start dulu sampai meiosis ni. Alright, thank you. Hope to see you again. Alright, class.